to learn the lymph, we need to have some basic knowledge here. We know our body is made up of various kinds of cells and tissues. If you take a cell contain cytoplasm, isn't it? And surround the cell also will find some fluid. Surround the cell also will find some fluid. We are not talking about the blood. We are talking about the fluid which is present between the cells. The fluid who is present between the cells that we are talking here. So, in the cell, whatever the fluid present in the cell, that can be termed as intracellular fluid. That can be termed as intracellular fluid. The fluid which is present inside the cell is called intracellular fluid. Okay, so that means in the cell. That we generally use it to say it as cytoplasm, and that only called as protoplasm. Likewise, these kind of words we use. We're going to learn more in cell biology. So here it is, intracellular fluid. Okay. Then what is this which is present outside? That outside between the cells which is present, that is called tissue fluid or extracellular fluid. We say that fluid as tissue fluid or extracellular fluid. Or we have some more names throughout the physiology that when you are learning, these names will come. So interstitial fluid, tissue fluid or extracellular fluid or interstitial fluid. Then we say it as a part of matrix or ground substance even. Whatever present between the cells we are talking about, between the cells, that fluid we say tissue fluid. Imagine if there is a blood vessel or capillary is there. So we'll draw that also. That make you can understand. This is a blood vessel. Okay, in the blood vessel, whatever it flows, whatever flow in the blood vessel, that can be called as blood. Okay, so that means to differentiate the various kinds of fluid, not much in number. So just we can classify basically in the form of three. That one is the fluid in the blood, that blood vessels, that we termed it as blood. Fluid in the tissues, we call it as intracellular fluid. Fluid between the cells can be termed as tissue fluid or extracellular fluid or interstitial fluid. Tissue fluid or extracellular. It's there in the name, you can understand. Extracellular, that means in between the cells, outside the cell. Interstitial, in between the cells. So likewise, the meaning will come. Okay. So this is one of the basic required for us to understand the concept of the lymph. And the one more, we need to know a little information about the flow of blood in our body, the circulation patterns in our body. So we know that we have heart which is four chambered. That human heart is four chambered with four names: right atria, left atria, right ventricle, and left ventricle. Right atria, right ventricle, left ventricle. That we have four names for each chamber. Right atria. So let me increase the size that you will be able to see. Okay, so what is this? That is here, this chamber we say left atria. So upper two chambers are atria. You may call it as auricles, no matter. That doesn't make much difference here, at least in first year. That actual difference we'll learn in body fluids and circulation chapter. Then this is right ventricle, and talking about the left ventricle. Need to talk about the left ventricle. Okay, so this is left ventricle. So there are four chambers in the heart: right atria, right ventricle, and left ventricle. From the left ventricle, we are not going here the complete details of the heart. So what we want, we are exactly that we have to learn over. So a blood vessel which arises from the left ventricle, which can be termed as left systemic aorta. that can be termed as left systemic aorta. In capitals, I'll make it left systemic aorta. Aorta or artery. Bigger arteries, we use it to say aorta. Okay, so for what it is? It is to supply the blood to the body parts. To all the body parts of our for all the body parts, this blood vessel is going to supply the blood. Okay, so whenever it is supplying the blood, so this particular blood vessel will be branched into capillaries. I'll show the capillaries, but before that, let me complete the circle, the circuit. Okay, so this is going to form the capillary. Smaller blood vessels can be termed as capillary. Okay, so these are all the blood capillaries, the smallest capillaries. Okay, so that means when the blood vessel is carrying the blood along with the blood, when it reaches to the body parts at body parts level, the blood vessel is going to be capillarized. We know these capillaries are made up of very thin epithelium, simple squamous epithelium. We call it as endothelium of blood vessels are composed of simple squamous element. So that is very thin epithelium, isn't it? Okay, so what is this? Let me complete this. Then after we'll go with the remaining issues. This is vena cava. Superior vena cava, inferior vena cava. So that means we are making a full round. That means arteries are on the way to supply the blood to the body parts. Then after that blood is collected by the veins that will come to the right atria. A journey from left ventricle to right atria. Left ventricle to right atria. In this mean period of journey, we'll find our lymph. So just at least in first year, you no need to remind the names here. If you remember, no matter. That will be much better, isn't it? So the veins collects the blood. The path is going to be like this. This is the path. So let me use another color. I'm using black here. So that you will be able to understand. So this is the path. Okay, so that blood will come to the body parts, and then after it will go back to the heart. Heart to heart. Left ventricle of the heart to right atria of the heart. That we need to learn. In the mean period of time, in the mean period of journey, what is happened that we need to go over. Okay, so before I continue, that for our easy understanding, I used to go for a small story which I used to say it as mini bus concept. After learning mini bus concept, we'll learn what is lymph. In future time, even if you are that want to remember the concept of the lymph, this story will be helpful for you. Okay, imagine from our college, in bus, need to draw the bus also. Okay, I'll try my level best. Okay, in the bus, nearly exactly hundreds number of students are going to somewhere else. That means picnic or something else. And I know in this period of time, going out is a dangerous thing. But just for example, hypothetical. Let me assume this is this year. This example is hypothetical example. Okay, so where we have to go? Let's go to where we have to go. Do you have any idea? Where do you want to go? You suggest one name. Picnic, tour. Any of the name you can give. Shall we go to Agra?
Wonderland.
Wonderland. Okay. So just what's there in your mind in that way? I am asking you. Okay, so one girl want to go Wonderland, but not suggestible this in this period of time, isn't it? Wonderland. Okay, so 100 number of people from the college, the Wonderland. College is organized the program, and 100 number of people in bus went to the Wonderland. Okay, all the 100 number got out from the bus. Everyone got out from the bus. Okay, you please imagine. I am little small. That I am not a bigger artist here. I can do something like boxes. Like I can't draw how people, 100 number of people, and how they are moving out. So among these 100 number of people, they went out and enjoyed in the wonderland. Okay, after a period of time, that was suggested actually up to a certain period of time. After that, you have to return back to the bus again. Okay, so some 85 number of people are entered in the bus. 85 number of people are entered in the bus, and the bus started back again to the home. That means to the place where it originated, into the college. Okay, so maybe you got worried over about the remaining 15 number of people who struck over in that. They're not came in time, isn't it? So that 85 number of with 85 number of people, the bus is started and moving towards the college. What about that 15 number of people who struck there, who are not able to catch the bus? For them, we are going to arrange a mini bus. Those who missed it there, for them, we are arranging a mini bus, that 15 number, 85 from here and 15 from here. These 15 number of people will join with our main bus before the bus arrive to the college. So that's why it seems to be nothing happened. Seems to be nothing happened. So whatever it is, before we arrive, before we enter into the main bus enter into the college, these 15 number of people with mini bus concept. That is, this is mini bus concept. We just remember here that 15 people are stuck there, not able to get into the main bus. So that's why we arranged a mini bus from the tour place, Wonderland. And then that before the bus arrive, main bus arrive the college, our mini bus able to catch the bigger one. So how many went out? 100. How many came in? 100. That's it. That is the positive ending of the story. Okay. So nothing happened. All 100 without corona get back to the college. Okay. Then next. Let us make these kind of comparisons here. That let us make. Why should you go forward? We'll have the heart diagram earlier. Let's go there. Let's see here. Okay, so that we have to compare with these particular issues. Imagine heart is the college, and 100 number of people are coming out from the heart. That means blood is coming out from the heart. Okay, so as I said to you, that everything will come out. That means all the 100 number. So now question raises here, why they are coming out and how they are coming out. Okay. So that means why they are coming out. For that we have answer. Why they are coming out? To supply the nutrients to the tissues. To supply the nutrient to the tissues, the blood contents will come out and bathes all the tissues and they supply the nutrients. Then they return back. Then they return back. Okay, due to the concentration gradient, we are able to catch up only 85%. Only they will return back here. Only 85% is returned back. The remaining 15%, for the remaining 15% fluid which is trapped here. So what is this? The fluid which came out from the blade. For why it came out? To supply the nutrients. So how much is returned back to the main bus? That is 85%. The remaining 15%. So to make up, to get the remaining 15% fluid which struck here, for them, we are arranging a separate capillary network, we say lymph capillaries, lymph vessels they become, lymphatic duct will join, that 15% will join with the main bus, that means the vein, before we open into the heart. Okay, a brief history of the story explained over. Now let me go for the original story. Now I am using with the technical terms. We'll go with the total story of this lymph here. Now you use the technical terms. I'm going to use the technical terms to learn all of this. So I'll make now the heart diagram. It's very small. So this is the heart. We just little minimize. Oh my God, utterly wrong because the atria are smaller and ventricles are larger. This is better. Yeah. Okay. So what we have to do? The blood vessel which is coming out from the heart, left ventricle, left systemic aorta. So that blood vessel. So the blood vessel is left systemic aorta, came out for what? To supply blood to the body part. To supply blood to the body part. When it comes to the tissue level, this blood vessel is going to be capillarized. Okay, so we call all these are capillaries, and all these capillaries are going to fuse and form vein, venules, then vein. So these are venules, and then vein. The vein will go back again. Vein will go back again to the heart. This is the story. So from here, we need to learn. So whenever the blood vessel is thicker, as I am showing in the diagram, capillaries are very thin. So these capillaries are going to supply it very thin. It is made up of simple squamous epithelium. So that's why when the blood with the greater speed, that means 120 mm of Hg, the pressure, which is from the heart, when it comes here, this blood immediately will come out from here into the tissues. Now we need to list out who is coming out, who is not coming out. So here in this case, WBC will come out. White blood cells will come out. The coming out of WBC from the blood capillaries into the tissues. Okay, we need to label this. What is this? Tissues. These are the tissues. So coming out of the WBC from capillaries into the tissues can be called as diapedesis. If you remember yesterday I shown in the text, diapedesis. The WBC coming out, we say diapedesis. Okay. Plasma will come out. Total plasma will come out. Okay. Some clotting factors won't come, but most of the plasma will come out. RBC are not coming. Keep that in mind. RBC will stay here itself. From the capillaries, RBC can't come. So that's why RBC will stay here inside. Only fluid is coming out. RBC stay, but WBC will come out. WBC coming out from the capillaries into the tissues called diapedesis. Okay. For why they are coming out actually? To supply the nutrients, to bath the tissues. So with the nutrients, they came out. Okay. Now let me say here, let me ask a question to you, that what is the color of the fluid which entered into the tissues?
Colorless.
Yes,
Colorless.
colorless. Very good. Very good. Why it is colorless?
Because it is white blood cells. No, sir.
White blood cells does not contain.
RBC is not coming out. But that RBC is not coming out means whatever the fluid will come out here, that is definitely colorless. Only who is coming out? WBC are coming out.
WBC.
Very good, very good. So you are following. Just ultimately we can say you are following. Even I am trying to confuse you. I made some red color here. Okay. So even you are not confused. That's very good. Okay. So the fluid came out. So the purpose is over. Nutrients are supplied. Then they have to return at capillary venular ends. Okay. So we have some specific names here. These are specific names, at least not required in this area. But when we are learning, let us learn full. Okay, so these are called arterioles. The branches will come out immediately from that artery is called arterioles. Okay. Next, in between, we say capillaries. That's the word we are using again and again. That is capillaries. Okay. Now, near the veins, whatever present, that we call venules. We say them as venules. Okay, so in general, we use it to say all are capillaries, but they have a specific name. Branches of artery is called arteriole, and after that, arterioles are going to be turned into capillaries, and all the capillaries are going to form venules. So, into the venular ends, they have to come out. Okay, they have to come in again. The work is over. All 100 students went to the wonderland, enjoyed. Now they have to come back to the bus. Okay, so why, how they come out? The pressure differences we need to create. So, this area now is going to show in microscope now for you. that box item, let me show you once again, the box item which I drawn there, that area, exclusively that area we are now showing in the microscope. Okay, so capillary, single capillary I am showing here. In this particular capillary, albumin protein molecules are there. If you remember, albumin, we didn't say it came out. Plasma protein, albumin. Yesterday I said the albumin I will say tomorrow when I explain the lymph. So, okay, RBC is also there in capillaries. So one capillary I am showing in microscope to please understand. Okay, so in between them, the Rolex RBC, our said RBC is there in between them. Then plasma protein, albumin, this is very important for us in the story. Okay. Albumin created high concentration gradient in the capillaries. In the capillaries, because of albumin, concentration gradient is created over. Okay, so concentration gradient. Shall we type this? High concentration. Albumin create high concentration. Okay, so then next, let's draw the tissues. As I draw the tissues earlier like this. Okay, in the tissues, the fluid is there in the tissues. Let me give some yellow color to the fluid. So the fluid is there in the tissues. So the concentration gradient in the fluid which is present between the in the tissues is low concentration. That so low concentration. Okay, just concentration difference make them our fluid to get back. So the concentration difference between these two, who created high concentration in the capillaries, the albumin protein molecule. So that's why the fluid is returning back. So because of the concentration difference, how much fluid is returned back, that we have answer. How much fluid is returned back? 85% of the fluid is returned back into the capillaries. Okay, so 85% of the fluid is returned back into the capillaries. So the story, in the story, 85% fluid is returned. Then now this fluid is moving into the venules to the veins. Okay. But what about that fluid who left over here after all 85% fluid went over here? So what about the remaining fluid which is not entered here? That means which is there still in the tissues. So for that fluid, we need to arrange some capillaries that we call lymph capillaries. Okay, so I'm drawing here the lymph capillaries. So these particular lymph capillaries, so these lymph capillaries, so these lymph capillaries, all the lymph capillaries fuse and form lymph vessel. Okay, then lymph vessel, all the lymph vessels will become lymphatic ducts. So that blind, highly permeable lymph capillaries will collect the remaining 15% fluid, and that 15% fluid is back to the vein, back to the main vein, and then this 85%, 15%. Again, we reach to 100%. So, what is the lymph here? What is the fluid? The 15% fluid, which is not written into the venue, which is not written into the capillaries, is called lymph. We need to say it as lymph. Okay. We have so many names. First of all, let me show you that names. Whenever the fluid present here, what do you call that fluid? Except RBC, that fluid is called blood plasma, isn't it? So whenever the fluid comes into the tissues, we have some more name. We say it as tissue fluid, or intercellular, interstitial fluid, or extracellular fluid. As I said to you, the fluid present between the cells is called tissue fluid, or interstitial fluid, or extracellular fluid. Okay. So this extracellular fluid, we can say. So whenever it is there in the blood, that is called blood plasma. So then When it comes out through the capillaries, at the time we called it as tissue fluid or interstitial fluid or extracellular fluid. Then after returning 85%, remaining who is entering into the capillaries, what it can be called? That can be called as lymph. Finally, we came out here to say what is lymph. Okay. So the blood, it is called blood plasma. Then in the tissues, when the blood plasma comes over, that is said to be tissue fluid or interstitial fluid or extracellular fluid. When it enters into the capillaries, lymph capillaries, collected by the lymph capillaries, that is called, considered as the green one. Everything green, whatever that I shown in the form of this capillary. This is a lymph capillary. The fluid in the lymph capillaries can be termed as lymph. Our mini bus. Simply we say, lymph is our mini bus. Okay, so what I shown to you earlier here, this mini bus concept. This is lymph, that fluid which is present in the tissues, which is not returned back into the capillaries. Okay, so before I continue with the lymph here, now we can go to the text and we can read everything. But before I go through that, yesterday that in we learned about edema, swelling. Interesting thing here that you can see that we have so many times we may face this particular issue. Edema, swelling of tissues. Why, when it will happen, that you can understand with the help of this here. So how much fluid is coming out? 100% fluid coming out. How much is returning back? 85% by capillaries, 15% by lymph. Okay, that's good. Imagine that how 85% is returning back. We have answer. Albumin protein molecules are created the concentration gradient. Okay, imagine if albumin protein molecules are less in number, if the albumin protein molecules are less in number, at the time what may happen? Instead of 85%, so I draw now the albumin less in number. So instead of 85%, imagine only 75% fluid is returned back to the blood capillaries. And remaining 15% is as usual, always will be collected by the lymph capillaries and they'll do their work. Okay, then how much fluid? Some 10% fluid will remain in the tissues, that which is not able to return back because of less number of albumin protein molecule. Importance of albumin I am giving now. Albumin protein molecule. So what happens? Fluid will be accumulated over in the tissues. When the fluid is accumulated in the tissues will come for swelling. The swelling of the tissues is called edema. You may heard over the swelling in the legs, that for some period of time, edema, that is nothing but edema. So edema is a swelling. Various reasons are there, like just like fever. That means fever, we have so many reasons. Plasmodium cause malaria fever, typhoid, salmonella typhi cause typhoid fever. Likewise, edema is a symptom actually. So how it happens, that's what I am saying here. Instead of 85%, if 75% or 70% or 60% is written, when will it do happen like so? So that albumin protein molecules are less in number. Okay, when the protein molecules are less, albumin protein molecules, because they are the reason for the concentration gradient they create and they make the fluid to get back in. Okay, so this is the storyline, and now let us let us find out that lymph issues here. Then after we'll go to the text here, and then we learn so how it is given. So understanding that lines, now it becomes so easier. So what we learn till the time, lymph. The participants in our story are lymph. Okay, lymph capillaries, which are blind, and blind, what it means, there is no opening to them. The blind lymph capillaries, highly permeable lymph capillaries, which is collecting that 15% fluid. Next, all the lymph capillaries opens into lymph vessels. Okay, so lymph vessels opens into lymphatic ducts. That lymphatic vessels, all the vessels opens into lymphatic duct, and all the lymphatic ducts opens into one lymphatic duct, that is left lymphatic duct or chyliferous duct, we use it to say. No matter, just lymphatic duct will write. And along with that, lymph nodes are present here. Lymph nodes. I need to show you the lymph nodes. That in the lymph vessels, when they are going like this, in between the lymph vessels, in some certain places, we'll find some swellings where our lymphocytes are going to say. Then generally it will happen at joints. Okay, so that means body joints. So there we'll find in that lymphatic vessel some sort of these kind of swellings. We call them as lymph nodes. We'll discuss more in immunology. So at least at present, lymph nodes are all participating in the immune system. Lymphoid organs like bone marrow and spleen are called as lymphoid organs. Including all of these, what do you say it as? We call it as lymphatic system. This is called lymphatic system. Lymph capillaries, lymph vessels, lymphatic ducts, lymph nodes, and lymphoid organs. We term this as lymphatic system. So this is the total story of lymphatic system. Now let's go to the text and let us see how it is given there. Lymph. Lymph is a colorless fluid, the property of the lymph. It lacks RBC, platelets, clotting factors, and large plasma proteins like albumin. It lacks RBC, platelets, and large plasma protein. But it has more number of leukocytes. And it is chiefly, that means every sentence we are making here, that the from the story, you can understand. But it has more number of leukocytes. What it means? We said from the capillaries, so WBC is coming out. WBC, leukocyte, nothing but WBC. And we named it as diaphoresis, isn't it? So that's why we can say that more number of leukocytes, and it is chiefly composed of plasma and lymphocyte. Lymph is chiefly composed of plasma and lymphocyte. When compared to the tissue fluid, it contains very small amount of nutrients and except fats, that because the lymph is the best carrier for the fat material. Digested fat will be carried by lymph itself. Then very small amount of nutrients and oxygen. Why? Because we came here to supply. So that's why when they are returning, definitely they'll have the less amount of nutrients, isn't it? So, but it has abundant carbon dioxide because it is collecting carbon dioxide from the tissues and other metabolites. What is metabolites? The waste materials, whatever formed in the activities of the cells. Okay. So the most important site of formation of lymph is interstitial space. What is interstitial space? The space present between the cells we call interstitial space. The space, here it is, the green one which I am showing. The space present between the cells is called interstitial space. As blood passes through the blood capillaries, now you try to imagine from our previous learnings. So as blood passes through the blood capillaries, some portion of the blood that include water, solutes, and proteins of the low molecular weight passes through the wall of capillaries into the interstitial spaces due to hydrostatic pressure at the arteriolar ends. Due to hydrostatic pressure at the arteriolar ends. That I have to show that for you. So that the diagram which we made earlier. So arteriolar ends. Okay, so these are the, now I have to use the color which we didn't use it till the time. Okay, so that let me go further. So this is arteriolar. Now I'm showing with the black color. Arteriolar end. So that is arteriolar end. The black, that sentence I've shown in the form of markings. Okay, so let me read once again. Most of the interstitial, that is fluid. Here it is, this word we read. Due to the hydrostatic pressure at the arteriolar ends. This fluid form the interstitial fluid or tissue fluid. Most of the interstitial fluid is returned directly. That means we said about 85% of the fluid is returned directly to the capillaries. How? That is due to the osmotic pressure in the venular ends. due to the osmotic pressure at the venular ends that is returned back. Now I am showing here how it is returned back. So venular end. Then how it is returned back? Concentration gradient. That is I explained now. Concentration gradient. This is concentration gradient. Because of albumin protein molecules, the concentration gradient is created. That means high concentration in capillaries and low concentration in the tissue fluid. This concentration difference makes over the fluid is returned back. How much fluid is returned back and from where to return back? At venular ends it is returned back. How much fluid is returned back? Nearly 85% fluid is returned back. Okay. So the most of the interstitial fluid is returned directly to the capillaries due to osmotic pressure at venular ends. Little amount of this tissue fluid, that means our said 15%, passes through a system of lymphatic capillaries like lymph capillaries of the intestinal villi, that they have the special name that is different, lymph capillaries and lymph vessels, lymphatic ducts, and finally reach to the blood through subclavian vein. Finally reach to the blood through subclavian vein. The bigger vein is the subclavian vein. Okay, so the names will come in next second year body fluid circulation chapter. So this near that vein that joined at the subclavian vein. Imagine this is going to be subclavian. Actually, subclavian vein is coming from our hand and going to the heart, collecting blood from our total four limbs. So that our lymphatic ducts are going to join with them to enter into them. The extracellular tissue fluid after passing into the lymph capillaries and lymph vessel, then the same fluid, that 15% fluid which is collected by the lymph capillaries is called lymph. Lymphatic system represents an accessory route by which interstitial fluid flows from the tissue spaces into blood. Accessory route, that I used this word mini bus, additional route, you can say. So especially these lymph capillaries in the intestinal microvilli, these lymph capillaries are called as lacteals. They are meant for absorption of these are the blood capillaries there. So our lymph capillaries is also present in the microvilli for what? For the absorption of fat, digested fat. We call them as lacteals. The lymph capillaries present in the intestine are called as lacteals. Okay, so that is going to come in the chapter digestion and absorption about the lacteals. Okay, so whatever, this is all about the lymph. So people feel so confusing, but no matter here. If you understand it as a small story, that means we are right there, learn the story script we are writing. That whatever we are reading here, the script of learned story, already we learned. So how it is given in the textbook, which we are reading like so. If you read like so, you'll understand the story. We know, movie we have already seen. That movie came as a novel. Movie I shown to you already. So this novel, when you are reading this novel, I don't feel so you'll feel problematic, isn't it? So that's it. That's the story of lymph. Okay, then that's the story of lymph.